it's Jose's turn now, and he's going to let us in on secrets of how to actually identify a mutation. I think very few of us realize how labor-intensive this is. We've heard earlier today from Fleur van Dijk how difficult it can be to be certain that you have the causative mutation for Marfan gene. Dr. Aragon Martin is an expert at finding these errors in what is a very long, complex gene. This summer, he's had the help of one of our summer students, Mr. Rohan Taylor, who has made a study of the most severe mutations found in the gene, and these are demonstrated in babies diagnosed at birth with the most severe type of neonatal Marfan. So we continue to map the gene, and José holds this map for the United Kingdom, and we also collaborate internationally with other maps, other holders of maps. So we're going to appreciate José's skill and appreciate the skill of other molecular geneticists around the world who actually share their results. José, can you tell us how you do it?
Yeah, sorry, let me just prepare myself. I was going to say as well that the project that we have from the summer student is going to be split into 2 years, this summer and the next summer, because it's a very long and complicated topic, which is for the neonatal Marfans. But today I'm going to talk about how to identify mutations. And just a little bit of history for our personal books. The Marfan Trust was founded in 1988. It was in 1991 when the FIN1 gene was discovered to cause Marfan syndrome. For many years, no UK lab was routinely screening the FIN1 gene in patients. And nothing changed until a couple approached Dr. Child and asked her what they can do to help for the screening of the gene in Marfan syndrome patients. And that is when the Sonalee laboratory was created in the year 2000, thanks to the many donations to the Marfan Trust. There has been many techniques used in Sonalee lab to screen the Marfan gene. Sonalee laboratory started with SSCA technique, which was very expert demanding because you have to identify, you have to differentiate between the bands of one sample and the bands of a control. This picture shows here on the right-hand side here will have been one of the easy ones. The difficulty came when the bands were too close to each other to observe the subtle changes. That is why Sonalee Lab started to use DHPLC technique, which also helped to improve identification of mutations in the Marfan population, but it also came with expert hands. So in 2012, the Sonalee laboratory moved completely to the technique called Sanger sequencing to fully screen Marfan patients, which is still considered the gold standard for mutations, for mutation detection. If you can spot typos while reading a book, I could have asked you to help me with the reading of these files. This is just to show you that this technique does not require an expert hand to read the sequences, but it comes with an expert hand to explain the findings. Since 2014, we have been learning how to use the next generation sequencing to identify mutations. And now, since 2022, we have moved completely to use whole genome sequencing approach to identify mutations in Marfan patients. While the other techniques will give you information in a specific part of a gene, in this case, the FIN1 gene, with the whole genome sequencing, you will get the information from all the genes in your body. All the DNA sequence in your body will be available to study. So, but how do we identify mutations? The DNA starting from the blood of a patient needs to be sent to an external company, which will run the samples in an NGS machine. By the way, I would love to have one of those machines in the lab, but the machines used in this whole genome sequencing are prohibitively expensive, so we normally send the samples out. Then we pre-process data from whole genome sequencing is sent to Sonalee Lab, where we process the data using very complicated computer programs generating a list of variants in each gene. This list of variants is very basic, so we add annotations to each variant using another computer tool. The basic area in this picture is shown inside a red rectangle, and the rest of the picture is part of the information taken after annotation of the sample. Just to let you know that there are about 170 columns of information from the different annotation tools. But I'm just showing here a portion of the column since it will be too big to show here. These annotations include things like the name of the gene, information about the variant, and other variant classification information. Perhaps the variant was already classified as benign or pathogenic by another tool, so this information will be available when we annotate the file. Once we have the annotated file, we first take a look at the information from the FEN1 gene to study possible mutations. To do this, we extract only the information from the known genes. These are the genes that are either associated with Marfan or related conditions. From this file, we will filter out the variants that have no priority, and we will keep those with priority. In this case, we will keep the variants found in the exons and splice sites, since there is a greater chance of finding a mutation in these two regions. The figure at the bottom is a picture extracted from the annotated file of a patient that have been previously negative for FEN1 using previous techniques. But just to clarify that Sanger sequencing was not done in this patient. It is very clear that one of the mutations takes our attention right away, and it is the mutation found in the FEN1 gene, the Marfan syndrome gene, which is here in this location here. All this information sometimes might be too much for the brain. For this reason, we can also visualize the mutation to confirm it with our own eyes. This is a picture of the mutation found in the entire FEN1 gene, all the mutations actually from this previous patient on the FEN1 gene. We will zoom to the region that we are interested, in this case, where the red arrow is located, which is the exon 42. In this picture, you can see the location of the mutation we are studying, which is this one here. It's the location here. And just to clarify, the letters of the DNA alphabet are A, C, G, and T. Is the DNA in the same? Is the DNA is the same as the reference sequence? So you can see the reference sequence here on this section. It will be shown as a gray area on the picture. So anything here, it belongs to the same nucleotides that you can see here. Any differences will be shown. Like, for example, you have a G here, and in this case, the mutation here, which is a T. Two gray lines. So this each line here, or actually two gray lines of belong to one cell. Oh, too wiggly. So yes, so you can see that there is a G in a red square here. This mutation belongs to one cell only in the participant's body, and therefore it could be either a mistake from the start of the screening or a spontaneous mutation in one cell. Since we are looking for mutations that started from birth, we are not going to worry about this G at the moment. And we're going to focus on the red arrow, which points to the location of the mutation. You can appreciate that some of the cells have a letter T and some have a letter C. It is very important to know the location and also the letter of the alphabet of the mutation, because a particular sequence of the DNA alphabet will produce a particular word, which will be translated to an amino acid. In this case, our mutation belongs to a word called CAG. This CAG will be translated to the amino acid called glutamine, which is the normal amino acid in the protein. But because we have a mutation in every cell, these cells will also have a different word. In this case, the word will be TAG. This TAG is the code word for stop what you're doing. Therefore, the message on the cells with the mutation will be stop creating the protein, the FEN1 protein. So the patient with this mutation will create half of the FEN1 protein that is required. And this is how we find the mutations. Sonalee Lab is looking, we're keeping a record of every mutation we found since the opening of the lab in 2000. We think this will be useful in the future when UK families request information on an old family member that was created with us to help with the mutation and screening. This is already useful because we have helped the NHS with many inquiries about present family mutations. Once we find a causative mutation, we also get in contact with a group that has created a database of FEN1 mutations called UMD, also known as the Universal Mutation Database. The previous mutation is very clear that will cause the Marfan syndrome in the patient, but sometimes it is very difficult because we need to do mutation interpretation to confirm the mutation as causative. I will leave this topic for another talk. The UMD is very important for us because we can always go to this database to confirm if our mutation has already been reported somewhere else. In this case, our mutation, this is a result of the analysis on the UMD. So it has not been reported yet in the UMD database, but it is okay, but because this type of mutation is normally pathogenic. Normally any stop signal are always pathogenic in Marfan syndrome. As an example here is the UMD report from a causative mutation found in Sonalee lab with the right link to that paper where we published the mutation. This is just a visual list of mutations found in the UMD FPM1 database and from our lab. The green lines over here on the top are the mutations found in the UMD database, and the yellow lines here on the bottom are our mutations, and the red ones are the variants that we found in the lab. And again, I just want to say thank you very much for everybody, for helping us to continue what we love doing, which is to find the genetic cause of Marfan syndrome patients and in other related tissue conditions. So thank you very much.
Marvelous. That was marvelous. Can you tell me, now that we have thousands of mutations around the world, is it true that each one tends to be unique?
I would like to say yes, because you can see that family members with the same mutation sometimes express differently the clinical features. Sometimes it's milder, sometimes it's severe. And also between families, they have the same mutation or the same location of the mutation, and they have different phenotypes. We think it's either because of the modifiers, gene modifiers, which are all the genes that helps to make it severe or helps to make it milder, the condition. And also we start to think as well that epigenetics is actually, it's nothing to do, it's something to do with your genes, but not really per se. It's like, for example, the body decided that I'm going to block this section here, or I'm going to block this section over there, and there must be a system there that is happening, and it might help as well with the severe or the moderate. There is a study on cancer where two twins were one of them had cancer and the other one didn't. So when they look at the epigenetics, they realize that the genes that the person has cancer, some of the genes were blocked by epigenetics, and the other twin who had didn't have cancer, the genes that were helping to protect her, they were available. So that's another condition we're trying to do. Yeah, we're trying to do that. So also, I was going to say that we're trying to do this genotype-phenotype correlation, where we can see patients looking at the variant in particular and see if what is the difference in between them. So we start to study that with one of the Rohan is actually helping me with the neonatal. We're starting with something simpler because we have a cluster and we can focus onto that. And in that cluster of neonatal, you can also see classical Marfans as well.
It must make your work much more difficult. For example, in cystic fibrosis, virtually all the patients all have the same mutation worldwide.
Yeah.
Makes life much simpler, doesn't it?
Yeah, we have the FPM1 gene, which everybody, I mean, I like to call any mutation in FPM1 Marfan syndrome, but you have mutations there that are associated with Wilmer-Cassani or other type of phenotypes, but I prefer to call it Marfan syndrome in the whole gene, and then everything is obviously different phenotypes here and there, which will be nice. What we're trying to do is classify those variants, so then in the future we can say, if somebody comes along and said, I got this variant, what is going to happen to me? Then we can say, for example, 60% of the people have aortic aneurysms, or we can say 5% of them didn't have, only have aneurysms. So we can probably help to manage better the condition.
Excellent. Any questions from the audience?
By the way, I'm always welcome if anybody wants to send the comments to my email. I'm always welcome to answer them. Don't be shy.
No, nothing's emerged on the chat feed. People have been debating the merits of Coca-Cola.
I was going to say that for mutations in the NHS, what they do normally, they send a sample to a panel and they screen the sample. So one of the help from us to the NHS is that we are doing research in the fact that they only look at panels, which is several genes. We're going to look at all the genes in the whole body, and not only the genes, the coding regions. We also look at the intronic, because like I said in my previous talk, sometimes the intronic regions, actually the intronic region that I was discussing before, it produces an insertion of a new exon, and that's what causing the cause of the Marfan syndrome. And yeah.
Jose, do you think we will reach a stage where people could contact you, and if they wanted to, they could actually pay for the gene testing themselves? Because I get contacted by people who say they're waiting so long, even just to get an appointment. Maybe it's a year for the appointment. Maybe the GP is not even happy to refer them for an appointment, and they really, really want gene testing. So how many years away do you think that will be? And how much do you think it will then cost people?
The good news is that last week, Illumina, who is the company who is making these machines to screen these expensive things, has been reduced slowly. Now they're saying, I mean, I said 200 pounds, but actually it's $200, which is probably up for the chain now, it's 170 pounds. So this is very good news, because I just hoping that when the baby pops out of in the clinic, it will come out with a USB key and said, this is my DNA, screen me, because we might need that in the future. And hopefully, like I mean, Dr. Chia probably better know than me, but I think we were screening before patients privately as well. The reason that I think we are very helpful is that at the beginning, we were the only lab who was testing the Marfan syndrome gene, and then we helped the NHS to with their own tests. And that, but although they are doing the test, we're moving forward to see if we can maximize the detection as well. So that's what we are focusing now on, to whole genome sequencing, while the NHS is still doing the panels. But I think it's because of the amount of money that you can put in resources. And I think, correct me if I'm wrong, Dr. Chal, we are collaborating as well with Deborah Rosendorf in Royal Brompton. So when she screened her families, and it's negative, we come along and help her to find out if there's another gene, or look at also the intronic regions, just in case the technique was not looking that area. But I'm just saying that we are so advanced now to find, because before we used to do just Sanger sequencing, which is just screening the gene, and MLPA, which will look for large deletions. Now with the whole genome sequencing, you can do wonders with this. So yeah. And then you don't have to do many tests. You just need to do one test, and then you can get the data for a long time and analyze the data for a long time.
Thank you.
Thank you very, very much, Jose. It's a fascinating topic.